you're going to watch a video of me doing a review of this for a class a couple years ago. So you get to watch all this again. And then make sure you do your reading, which the reading section for tomorrow is real short. It's like 2 pages. It's about a chromosome. Remember when I showed you the video of the chromosome coiling up?
Yeah.
That's basically what the last reading section is. And we've already talked about it. So it's a good time for a review. And I'm going to be gone, so it all works out.
So another AP seminar, or is that what it is?
Yeah, it's another AP workshop we're going to.
What? Do you need my thing?
Yeah, I'm going to keep your book. Do y'all have a lab book you want to loan me?
I do. Another book. I will.
Yeah.
Are we supposed to need our books? Those red books?
If you have a red book you want to loan me, I can let the teachers down there see it. It's kind of helpful for them. You what?
This is such an awkward angle.
Hello, your border still says everything. DNA molecule, telescope, all those magnets. Tell we're in science class.
What? He was like, who was it? I guess.
Please come again.
Please come again.
So how long did it take you to write these PowerPoints? Does it take you a while? Every year you...
They have a template that I use.
I see.
It's not too bad.
It was fun with the guys at school.
It's some work, though.
It was really fun. You can say anymore? Like in Spanish. Like you say
Okay. This is the transcription process. And what page is this in your book?
225.
225?
First step, transcription.
So we already talked about this, but I'll review it. And so here we go. Basically, a enzyme called RNA polymerase attaches at a site on the DNA called the promoter. It goes down and kind of unhooks the DNA. And then one by one, it puts on RNA nucleotides onto the DNA template strand. And that's a good way to say it. It hooks RNA nucleotides onto a DNA template strand. And it hooks them on. Hook. Hook. Click. And eventually it'll form this strand of RNA that we call what? Do you remember? mRNA or messenger RNA. Now, if this were TCAAG, then what would this be? Don't yell it out, write it down. If this were TCAAG on the DNA, what would the RNA be? Just write it down, don't yell it out. Taking you this long to write something down, it's probably not ready. You know it?
I think so.
Oh, wait, don't say it. I want everybody to say it together. We're all going to try and say it at the same time, okay?
Oh, wait.
So we're going to start here and go to the right, and we all try to say the letters at exactly the same time, so it sounds in unison. A good class could pull this off. Okay, you ready?
A-G-U-U-C.
A-G-U-U-C. That's correct. Because RNA has uracil instead of thymine. That was good. Y'all did well there. A couple of y'all off, but that's okay. So, what's another difference between RNA and DNA?
Single strand.
Has ribose instead of deoxyribose as the sugar.
Single-stranded.
It's single-stranded instead of double-stranded.
It's not a helix.
It's not a helix. So this thing will detach, and it will make its way off, and the DNA comes back together. We conserve the original copy, and eventually the RNA polymerase falls off at the terminator.
When does this happen?
This happens inside the cell, inside the nucleus. It's constantly going on as the cell's making new proteins.
Can DNA like replicate stuff while it's doing this?
No, actually, when DNA is replicating itself, it'll stop doing this. Yeah. Question?
Wait, if it already has the RNA stuff like together, then why does it need the DNA?
It doesn't have the RNA together.
So, but what
it's
is
putting the RNA together from the DNA.
What does it do exactly?
It's basically copying the DNA. Now we have a copy of the DNA.
Where does the RNA stuff come from then?
The RNA, that's a good question. The RNA is balled up here. It comes from the nucleolus. Do y'all remember that little ball that's inside the nucleus called the nucleolus? That's RNA. And it's got a bunch of RNA nucleotides that can be used to do this. Where it originally got the RNA nucleotides, there's different ways. Some cells can make them up from scratch, from glucose and other things, and changing around molecules in the Golgi apparatus to finally make this.
But why does it need the DNA? Like, what is it?
The DNA is the code that we're going to use to make proteins.
I mean, like, how does it work exactly? Like,
I'm showing you how it works right now.
I mean, does it just like does the RNA already have like the RNA on it, or I mean, the polymerase, does it have the RNA on it, or?
No, these RNA molecules are floating around here.
So they just like hit it
and bumping into it, and if the right one bumps into it, it'll stick it on.
So they just keep hitting it until it fits.
That's right.
Okay.
Good. Yeah. These molecules are floating around bumping into things. Good job, Cole. Alright, so here is the RNA, and we have to do some things to the RNA before it can leave the nucleus. And we call this piece, again, we call it messenger RNA or mRNA. And before it can leave the nucleus, did you read about what happened?
The introns are spliced off.
That's correct. What we're going to do is we're going to do something called mRNA processing, where this strand of mRNA is changed around a little bit. And for that, I'm going to use my long mRNA model. Okay? That we got to do a couple things to this before it can leave the nucleus. The first thing, we're actually going to do three things. The first thing we're going to do is we're going to put a cap on it. Every RNA needs what's called a guanine cap. Cap. A guanine it's a guanine nucleotide. It's a G. So it's capped with a G.
What is cap? I don't know if it's an RNA strand,
The guanine.
It's a guanine.
Well, that's what
but
RNA doesn't normally have.
No, the thing on the end.
No, it's fine.
You're just putting it on there.
RNA.
For the RNA
nucleus.
mRNA strand has capped with a guanine base.
Nice, man. Make it rainbowy.
How do you make it not rainbow?
Yeah.
Mr. Wells tried for an hour, couldn't figure it out.
We can't figure out how to fix it. So you put a guanine cap on one end, boop, and on the other end you put what's called a polyadenine tail, which is adenine nucleotides, one after the other. Bam, bam,
What does a cap do?
bam. A guanine cap and a polyadenine tail. When I say poly, I mean P-O-L-Y, poly-A tail. Poly means many. So many adenines are added to the tail.
Why didn't they have to be added?
They think the poly-A tail helps determine how long the mRNA will live in the cytoplasm. The longer the tail, the longer this molecule lasts out here in the cytoplasm.
Does like adenine not close it off like guanine does or something? So
I don't know why. They're trying to figure this out right now. This is ongoing investigation. But all you need to know is that you had a cap, a guanine cap on one end, a polyadenine tail on the other, and then you're going to snip out some pieces of mRNA. Some of these letters aren't used. Like there might be a sequence in here of 50 letters that aren't going to be used at all. And so we're going to snip them out. And there are special enzymes that snip out these pieces, these unused pieces of mRNA. And we call an unused bit of mRNA an intron. And again, this is a very long strand. It's longer than my model here. And so there are many, many introns. And the part that is not snipped out, that is left, is called an exon. So the introns are snipped out and the exons are left there. And I'm going to show you a video of how that's done. It's kind of funny. The enzymes that cut out these introns are called SNRPs. It's kind of a funny name, which is an acronym that stands for something.
Is there another word you would use for snipped out? Like, is there
Here it shows spliced out. Yeah, spliced is removed. So here we go. Here's the piece of RNA. It's called pre-RNA. And there's the cap is put on one end. The poly-A tail is put on another end. And these little spliceosomes come in and they cut out the introns. And then all that's left is the exons. And so the final piece of RNA is actually shorter than the original piece of RNA.
What happens to the introns once they're cut out?
I don't know. They're probably recycled and used again, cut back up into their individual nucleotides and reused.
Real quick, what are those two symbols? Oh, the 3' and 5?
3' and 5', yeah.
And I've kind of forgotten what 5' and 3' are again.
The 5' end is the end that the phosphate hangs off.
Okay.
And the 3' end is the other end. So this would be the 5' end, and this would be the 3' end.
3' and 5.
Okay?
The 3' end is where
I got it backwards.
The 3' end is where the
The tail goes on the 3' end. The cap goes
what
on the
end of the nucleotide is 3'?
The 3' end is the top of the nucleotide, as this model is. The 5' end is the phosphate end, the bottom.
Okay. 5 is phosphate.
So here we go. Here's a video showing how spliceosomes work.
Most eukaryotic genes are composed of numerous short coding sequences called exons, interspersed between long stretches of non-coding sequences called introns. The initial messenger RNA molecule copied from a gene includes introns as well as exons. Before this RNA molecule can be used to make a protein, the introns must be removed by a process called Rna splicing. A structure called a spliceosome plays a key role in RNA splicing. The spliceosome is composed of several small molecules called snRNPs, or SNRPs. SNRPs are made up of protein and small RNA molecules. Formation of the spliceosome begins when one kind of SNRP binds to the 5' end of an intron, and a different kind binds to the 3' end of an intron. Additional SNRPs then interact with the complex, bringing the two ends of the intron together and causing the intron to form a loop. This also gathers all of the SNRPs together as the completed spliceosome. The intron is then cut out and the exons are joined or spliced together. The spliceosome then falls apart as the SNRPs are released.
Huh. Isn't that cool?
Does that just take out the intron
Takes out the introns.
Snurps take out the intron.
Disappear and float away.
The intron or the snurps?
The intron.
Yeah, the intron, I don't know what happens to it. It's probably recycled. Probably torn apart and the
Wait,
nucleotides
what's the
reused.
difference between spliceosomes and snurps? Yeah.
A spliceosome is all the snurps combined. A spliceosome is made of 5 snurps.
Got your name and was a genius.
Alright, so what does an intron do?
An intron is a
An
non.
intron is an unused bit of information.
That's just, okay.
What it is, it's left over from evolution. I was explaining, Stephanie came and asked me this earlier. For instance, you know, birds came from dinosaurs, right? You've probably heard that before. Well, birds have dinosaur information in their DNA. But the dinosaur information is not used, so it's cut out, you see. And only the bird information's left. So there are traces of past organisms in your evolutionary past in your DNA. And those can be read. And it's more proof that evolution happens, and you can actually tell what an organism comes from because you can see genes from previous organisms in their DNA.
That makes sense.
Yeah.
So we have like unnecessary like primate or
Lots. And there was actually a funny Star Trek episode where the introns became active and the people started turning into lizards and old little mammals, and one of them turned into a spider, which makes no sense because we're on a different evolutionary path. But it was good for the show. Okay, so now finally, after we have this final piece of RNA, mRNA, and it's been all processed with caps and tails and introns cut out, then the whole thing can leave the nucleus through one of those nuclear pores and can make its way to a ribosome. Here's a ribosome. Now, a ribosome actually has 2 what we call subunits. A large subunit and a small subunit. Y'all remember ribosomes, right? The little dots in the cell? Do you remember? You probably memorized the function of a ribosome. What was it?
They make
Synthesize
proteins. They
proteins.
make stuff hard. Like, they make proteins.
They make proteins. Well, that's what we're trying to do here. We're trying to make a protein. I'm going to show you how that's done now. Aren't you excited?
Super.
Jonathan, are you excited? You seem very much. Okay, so here we go. The mRNA goes into the ribosome. Now, a ribosome is also made of RNA. But it's RNA, it's a strand like this, all folded up, and also some protein. So a ribosome is made of RNA and protein. So we actually call this piece right here, we call that mRNA. And this ribosome we call rRNA, because it's also made of RNA.
Ribosomal.
Ribosomal RNA. And this is messenger RNA. And there's some more RNA out in the cytoplasm that you have to know about too. These little green things here,
tRNA.
they're called tRNAs.
Transfer.
Or transfer RNAs. And they're made of RNA too. So it's a strand like this, all coiled up in a certain way. It's actually coiled up to have sort of the shape of a cross or a letter T or something, or just some weird symbol. This is actually a long piece of RNA all coiled up, and it's called tRNA. And what tRNA does is each tRNA carries an amino acid, which, if you remember, amino acids make proteins. Right?
Each RNA carries amino acids.
Each tRNA, that's this green one I have up here, carries an amino acid with it. Let's use the yellow.
How does a strand of RNA turn into that?
Turn into this?
Yeah.
There are enzymes in the nucleus that shape it the right way as it leaves. Turn it the right way. Enzymes do everything in the cell. So there's an enzyme that grabs this long strand of RNA and shapes it into a tRNA and lets it go.
What was an enzyme? A protein, was it?
An enzyme is a protein. That's what we're trying to make. We're seeing how the enzymes, the proteins, are made. Yes?
Oh, wait, what does the transfer RNA do?
The tRNA carries amino acids.
What are the white things?
That's all it does?
That's what it does. I'm going to show you. It does more in a second. I'll tell you what the white things are in a minute. Do you remember what a protein is, though? Mm-hmm. It's a chain of amino acids. So we're going to hook these amino acids together to make one of these. And then this will fold into the final protein shape.
Quaternary, tertiary, secondary.
Primary, secondary, tertiary, quaternary structure. And the quaternary or tertiary structure, that's the enzyme. So we're making enzymes here. Once we have enzymes, we can do anything in the cell. So, here's how it works. The mRNA kind of docks inside the ribosome. And the mRNA is read 3 letters at a time.
Triplet something?
Yes, we call 3 mRNA nucleotides, we call that a codon. This first codon that I have here is AUG. AUG is also known as the Start Codon. Those are the 3 bases I have here. You can't read them from where you're sitting, but trust me, it's adenine, uracil,
Always?
guanine.
Are those 3?
Yeah, AUG is always what we call the Start Codon. And then what happens is these tRNAs start floating in here. And the tRNA has attached to its base a codon. It's not actually a codon, we call it an anticodon.
So now that that's in there, it's now an anticodon instead of a regular.
Well, it's always an anticodon anywhere. But that's what we call it. The bottom 3 bases on the tRNA is called an anticodon. Now you have to understand this tRNA is a lot bigger than this model. But anyway, the tRNA with its anticodon comes into the ribosome and it tries to fit with the codon. It tries to match the codon. So if this codon is AUG, what anticodon would match it?
TAC.
Well, it's not TAC because there's no T in RNA.
UAC.
So UAC would match AUG, wouldn't it? Well, this isn't UAC, so that doesn't match. So this is the wrong tRNA, and it'll leave. And then another one will come in and test itself. And that doesn't fit either, so that leaves. Whoops, I want to switch these.
What are the white things on the tRNA?
That's the anticodon. Those are bases that match up with these bases.
They don't have like a...
They're nitrogen bases. They do. They have a sugar and a phosphate with them. But I didn't have room for that in my model, so I left it out. Is that right? So here, look, this tRNA comes in, and yeah, it's UAC at the bottom. So click, it fits in exactly.
So what happens?
The Goldilocks.
Yes, that was the right one.
So what happens now?
So what happens?
That's great. I'm going to do that now. Everybody with me so far? How would I say this if I was saying it scientifically? The tRNA enters the ribosome, and if its anticodon matches with the codon of the mRNA, Then it will attach. And the next codon is then exposed. Ooh, the codon is exposed. And more tRNAs will come in and try to match with the next codon. That one doesn't match. That one does match. It took me hours to get these things to match.
So 3 nucleotides in an mRNA are a codon.
That's correct. Very nice. Well said.
3 nucleotides in an mRNA.
No, like on the mRNA strand.
Oh, on
3
the mRNA.
nucleotides.
Mhm. 3 nucleotides make a codon. It's actually 3 nitrogen bases, but each nucleotide has 1 nitrogen base. So that's correct. Now 2 of them have docked. And then what's going to happen is the 2 amino acids are real close together, and they will form a bond between them. It takes ATP to make this bond. So this whole process uses energy.
How much ATP does it take?
We call that a peptide bond. It takes 1 ATP. It takes 1 ATP to link these 2 together. And then they're put together. It's called a peptide bond. A peptide bond is a bond between 2 amino acids. And guess what, y'all? We got the first 2 amino acids joined together of our chain. Can you guess what happens next to finish this chain?
Keeps it working.
What happens is the first tRNA that's carrying this first amino acid, it leaves. But it leaves its amino acid, you see. And then it goes back in the cytoplasm where it will dock with an enzyme that puts on another amino acid. The same kind that it left off in the first place. So this tRNA only carries the blue amino acid.
So is there some kind of correlation between like the anticodon and the amino acid it has? Or?
That is an awesome statement you just made, Cole.
Is it like,
I very much like it.
Can you say it again?
He said, is there some kind of correlation between the tRNA anticodon and the amino acid it carries? Yes, there's a very definite correlation, which is on page 224. Here's the correlation.
Oh.
You didn't know that?
Where's that tRNA go?
Hold on. Here's the correlation.
My calculator.
Are you all right, Trna? Oh, you're showing it. You can show it up here.
But it's rainbowy.
Oh, okay, because it's rainbowy. Okay, you can go to the other one. Whatever you do, you're in charge. So this is, Cole, this is the correlation not between the anticodon and the amino acid it carries, but it's between the codon and the amino acid, which is still a correlation. And so you can see, here's the start codon, AUG. That codes for the amino acid methionine. So wherever you see AUG in the codon, you're going to end up with methionine, which, look, I've made it, METH, meth.
So
Is that a type of protein?
That's a type of amino acid.
I know, why does that
Is
even
it?
matter?
Because when you put all the amino acids together, you got a protein.
I know, so a protein is made of a bunch of different amino acids?
That's right.
Made from the RNA. Made from the tRNA.
Yeah, with the tRNA brings in the amino acids.
So it's just like transporting it.
That's right. That's why it's called tRNA transfer RNA.
Oh.
Good job. Yeah.
So is methionine?
Wait, no, it was Hannah.
Oh, sorry.
When that green, like, pea thing, like, what makes it...
tRNA.
What makes it just go away after it, like...
Because these are molecules, and molecules are always moving. And so it's just randomly flying around until, whoop, it goes into a ribosome, and if it happens to stick there, it's stuck there.
But what makes it...
And then when this bond is made, it breaks free and leaves. And And it's all happening very fast. This goes at 15 amino acids per second.
This is really fast.
About 15 amino acids per second are put on here. So these molecules, Hannah, are just flying around, and maybe they happen to doop, and then boom, after their amino acid is bonded. This whole ribosome is an enzyme that catches for a minute and then releases. So it's like it's held there for a second and then let go. Yes?
So is methionine always like the first in the chain?
Methionine is always the first codon.
Oh, wait.
It's the
So
start
it's not
codon.
the first.
The first amino acid.
In like every chain?
In every protein, the first amino acid is methionine.
Pretty cool.
How many amino acids are there?
There's 20 different kinds.
So that's just one of them.
And so you can see this whole chart has all 20. AGA is arginine. LEU is leucine. Where's LEU? Always like leucine.
C category on the left.
C,
Right.
leucine. There's leucine. CUC, CUA, CUG. All code for leucine.
What are the stops?
Well, we're going to get to the stop. We haven't got there yet. Okay, so this tRNA that was holding methionine leaves, floats away, and it will eventually run into an enzyme that's carrying another methionine. I don't have another methionine handy, so I'll use this one. But another blue one, and it attaches it. And so this one's ready to go again. It can go back into another ribosome and drop off its methionine. Okay?
So it doesn't go back to the same mRNA?
It could go back to the same one later, if there's another AUG somewhere.
So basically they just like dock once and then head to the next one?
Yeah, they're just delivery boys. They deliver the amino acid and then they leave. Emery's is boring you?
Yeah.
You seem to have your eyes closed. Okay, so after the tRNA leaves, the whole ribosome moves a little bit. It slides down. So ribosomes are moving while this is going on. And another tRNA will come in, dock, another peptide bond is made. The first tRNA leaves and goes and gets another green. If there was another green up here, I'd put it on there.
So the second one stays until another.
Second one stays. And then the whole ribosome moves down again. And then the tRNA comes in and docks and bonds. This one leaves, goes and gets another yellow, and the whole thing moves down again.
Like a root.
And then another one comes in. This one doesn't fit. I lost one of my tRNAs in the years. Attaches. This leaves, goes and gets another pink. The whole thing moves down. And that will eventually continue again at about 15 amino acids per second until you finish your protein, which is usually several hundred amino acids long.
Cool. How many is it? Sorry, you just said it's not
About several hundred. Several hundred. It can be anywhere. There are some short proteins with 40, 50 amino acids, but most of them are 300-400 amino acids long. Here's a good question for you. If I have a protein that's 300 amino acids long, how long was the DNA code, how many nucleotides long was the DNA code that coded for that protein?
300.
Wait, how many?
300.
300 amino acids long.
300.
How many nucleotides are the code that coded for a protein?
42.
900. Because each amino acid has a 3-letter code. Right?
U, all that.
Well, that's true. It would have been longer than that if you consider the exons. Do it, Hannah. You're thinking, I like that. Actually, it would be long if you consider the exons. Prokaryotes don't have exons, so let's pretend we're in a prokaryote. They would put that in the question.
Okay, so three amino acids.
3 amino acids is three nucleotides is a codon. acid is coded for by 3 nucleotides.
So you're asking how long the DNA was?
So how long was the DNA that coded for a protein that's 300 amino acids long? 900 nucleotides, or 900 nitrogen bases, which is 900 nucleotides. So you know what we call this whole thing from promoter to terminator, including the information in the middle? A gene. That is a gene.
Like Eugene?
No. A gene. So on your DNA is just gene after gene after gene for making protein after protein after protein. You have about 20,000 genes on your DNA.
DNA is gene.
That's what DNA is, a bunch of genes. Yes?
They're just proteins, though.
They're proteins.
But they give us characteristics, right?
That's correct.
So how does a protein give us black hair, red hair?
Well, for instance, if you have a dark colored protein in your hair is going to be dark. If you have a light colored protein in your hair is going to be light.
I see.
If you have dark colored proteins in your eyes, then you have dark eyes.
So we
eyes.
see the proteins when they're looking at color?
Proteins
give color?
Proteins end up giving color, yeah.
Okay.
Cool.
Yes.
Can you use a Punnett square with this? Or
Yes, because the gene is the allele. So in other words, pea plants had a gene for tallness. So this gene is the big T. It's the little letter that I put on the chromosome. If I put big T there, that's the tall gene. So somehow that pea plant is making proteins that end up making it tall. I don't know how that works. It's different for every organism.
Yeah. Every organism's DNA is different, like that.
That's correct.
It's just different. I see.
That's correct. My DNA is different from yours. That's why you look different from me. I have more short genes. You have more tall
Mac and
genes.
Lee have the same genes.
Yes. We do.
Yes. All their DNA is the same.
Yes.
Except for some mutations that might happen. That's the only thing that can make them different.
Fingerprint in my ear.
Okay, so.
And your tongue breaks so
Was your
different.
ear were you born like that, or is it something else?
No.
That's what they told us when they first came here.
No, I was born like that.
I believe it.
I was born like
Baby,
that.
I was born this way.
Lady
Lady Gaga.
Gaga.
Okay, let's finish. We're not done. So the last codon is the finishing codon. It's called a stop codon. And it's always one of 3 things. And UGA is one of them, so it's easy for me to remember. UGA, University of Georgia, stop. Stop with all the Georgia craziness. I went to Georgia Tech. So stop, Georgia. No. But anyway, that's easy to remember. Also, UAA and UAG are all stop codons. And so what that codes for is a special type of tRNA called a releasing factor.
Are we going to need to remember any of these?
Yes. You need to remember all of it.
Yeah.
Wait, so what's the pink thing? The stop codon?
The stop codon, this is called a releasing factor that comes in and attaches to UGA, and it'll attach,
You just said this.
and the first tRNA leaves, but there's no bond made to this stop factor because it's not carrying a amino acid. So there you go. There's your protein. And what will happen is the ribosome will kind of come undone like this, and the releasing factor will leave, and there you got your protein just floating out there in the cell. And then your protein will fold into its final formation, aided by special folding enzymes, folding proteins that get it to fold right, which is a whole other subject, which we won't get into in this class. Yeah.
Okay, I'm confused with the difference between stop codon and then the releasing factor. Are they the same?
The stop codon are the letters UGA
On
the-
the RNA?
on the RNA.
And then, so then
The releasing factor is a tRNA piece that binds to that,
okay, fine.
And-
to the stop codon.
binds to the stop codon, and causes the tRNA to leave without another bond forming, so that the protein is just left there floating in the cell. Yes, Hannah?
Isn't the end always supposed to be a G? Or like there's always like a cap or whatever?
Yeah, you're right. That's not red. The cap or whatever is, or the tail, let's say it's the tail here is hanging off. That's not, the tail won't be red. The stop codon comes before the tail. And what they think happens is that tail, that poly A tail, they don't know this for sure, but this is a guess, that maybe one of the A's is knocked off every time it goes through a ribosome. So that this mRNA can go through a ribosome 50 times, making 50 of these proteins, and after that it's done, and the thing will be destroyed.
How does this stop?
That's a guess of what the tail does. As far as I know, and maybe they've already figured it out and I just hadn't read about it, but as far as I know, they really don't know how the tail works. But it does help determine how long the mRNA will last. See, this mRNA can go through another ribosome. Yes?
How does it destroy itself after it's, or how does it die?
An enzyme eats it up.
Oh, okay.
Some enzyme will be there that's the mRNA destroying enzyme, and it'll tear it apart. Yes, Caitlin?
So those last three, don't get to...
They don't get rich. No, they don't get, no amino acid is made for them.
So they just
They're just there for another reason, determining how long the mRNA lasts. How long this lives inside the cell.
The stop codon is also UGA.
No, there's three though. See up there? UAA, UGA, or UAG. Yes?
the tRNA is the releasing factor that attaches to the UAG or UAA.
Okay, these are all tRNAs.
Yeah.
Some tRNAs bring in amino acids, but there's one type that binds to the stop codon.
That's called the releasing factor.
It's called a releasing factor.
How does the stop codon know when to come in, or is it just randomly?
It's randomly coming in all the time, but it won't connect with anything that's not a stop codon. It'll only connect, this one will only connect to UGA. So it'll float into the ribosome and come off, float into the ribosome and come off. And if it floats in and hits UGA, then that ends the protein. Molecules are always moving. You have to imagine this in a cell. These molecules are just flying around everywhere.
Ow.
Yeah, that's what would happen if one hit you and you were that small. Yeah.
So that's just examining the chart. I see for example, valine has GUG, GUA, GUC, GUU, but there's still valine. Is that what it is?
All of these codes code for valine.
I see.
So GUG, if the letter is GUG, you'll get a valine. If the letter is GUA, you'll get a valine. Valine has many codes for it.
Did you have to memorize all these when you were...
No, but you have to be able to use this chart. Like they'll give you a series of letters. You might see this on the AP exam.
You just start with the first three.
UAG, CCU, AT, AUA.
But the UAG
And you have to tell, using this chart, you have to tell what 3 amino acids will be there.
Well, in UAG, wouldn't that just stop?
Oh, sorry. So yeah, you'd stop it right at the beginning. That would be no good. How about
UAC.
UAC? So what does UAC give you?
Tyrosine, I'm sorry.
Tyrosine. And what's CCU?
CCU.
That's proline.
Proline.
Where is that?
What's AUA?
Isoleucine.
Isoleucine.
Leucine.
Oh yeah.
First letter, second letter at the top, third letter on the right.
So that means we have to memorize what 20 amino acids of the names are?
No, you don't have to memorize any of this. You have to be able to read this chart.
Read the chart. Oh, I see.
If I could give you on the test a sequence of letters and say which is the correct code, which is the correct order of bases, and so you'd have to read, you might have 4 choices, you see?
I see.
Check this out. What if there was a mutation?
Oh.
A mutation that changes this C to a G.
And you raped the wrong person.
It stopped.
It would stop. And you wouldn't even get to these amino acids. It would shorten your protein.
That person would die.
Well, you'd have a messed up protein.
Oh, it's not.
Now, it depends how important that protein is. If it's a protein that's not very important, it might not have much effect on you. If it's a protein that's really important, you could be really screwed up. It could be a protein that has some new function that makes the organism even better. That's how evolution works.
How do Siamese twins work? Or conjoined twins work?
That's something different. It's when the egg starts to split, like it did for you two guys, but it doesn't come all the way off. They're stuck. It stays
So
stuck together.
it doesn't go through telophase. Or
Doesn't go through cytokinesis correctly.
and
And so the two both grow to a person that's stuck together somewhere.
What about Siamese?
that can cause
That's the same thing.
But why?
Conjoined twins. It was named after Chang and Ang Bunker, who were from Siam, which is now not in Thailand. Okay, so that's